My name is Ryan Leung, and I'm a third year medical student from the Western University of Health Sciences. My project sought to review the current literature on the gut microbiota's influence on taste perception and eating behavior. So, why taste in the first place? Well, as we all know, obesity is a major disease in this country, increasing risk for heart disease, diabetes, certain cancers, as well as a whole host of other complications. Taste and the hedonic reward we gain from food plays a major role in food choice and also the habit of overconsumption. Well, what is taste exactly? It's one of our five senses. It's the way we sense chemicals and nutrients we ingest both on our tongues and, importantly, in our gut as well. These nutrients are then relayed in combinations of salty, sour, bitter, sweet, and umami. And the jury's still out on whether fat is a taste or not. So, if taste influences our behavior, then this begs the question, does anything influence our taste? Well, the gut microbiota has emerged over the past couple of decades as having critical functions in both health and disease. So this project seeks to explore that relationship in three key areas: associations between taste and microbial signatures, represented in the table right here, probable mechanisms of influence, and clinical correlations of pathogenesis, diagnostics, and therapeutics. The focus of this project was placed on articles published after 2010, and we also chose to focus on research that explored changes in taste perception rather than taste preference, as taste preference involved another layer of influence through the dopaminergic reward pathway. This table represents how the abundance of certain microbes were associated with changes in taste perception. I just want to highlight a couple of key associations that can help us anchor us to the different mechanisms. Lactobacilli, known for producing hydrogen ions, was associated with a decreased sour taste sensitivity. So the idea here is that the baseline production of hydrogen ions produced an acidic environment and likely lowered the threshold for sour. Another key experiment here, germ-free mice being associated with an increased preference for fat. So the gut microbiota is necessary for optimal energy extraction, and since this energy extraction was decreased, the mice likely had different compensatory mechanisms to have them ingest more fat. And these were found through an increase in lingual fat receptors, leading to them eating more, and a decreased fat receptors in the gut, leading to dampened post-ingestive satiety signals. So now onto the theoretical mechanisms. So the papillary structure of the tongue actually forms a pretty unique ecological site where its numerous depressions can encourage a buildup of biofilm on the tongue. And what this biofilm can do is act as a physical barrier, preventing and dampening tastins from reaching their proper taste receptors. This has been supported with certain tongue scraping studies having patients have an increased taste sensitivity after doing these tongue scrapings. These bacteria can also produce bioactive metabolites like short-chain fatty acids, bitter precursors, and organic acids that are really close to the receptors, suggesting a likely interaction. As explained before, there's a receptor expression model that metabolites can alter taste receptor expression, either upregulating or downregulating them. And these same taste receptors that are found on the tongue are found in the gut on enteroendocrine cells. So this also leads another avenue for influence. Represented in red here is an inflammatory model where we have LPS from bacteria activating toll-like receptors and releasing cytokines. Now this can happen locally at the taste bud level, and it can also happen in the gut, where the release of cytokines will enter systemic circulation, causing systemic inflammation, which has been known to influence taste perception. Short-chain fatty acids produced by bacteria and LPS can also activate enteroendocrine cells, and influence the release of certain satiety hormones that enter systemic circulation and again have been known to influence taste perception. Lastly, it has been hypothesized that bacteria may directly trigger the vagus nerve, which is responsible for nutrient sensing. Dysbiosis is characteristic of all of the pathologies described in this section, and the mechanisms that we previously went over might help explain the different gustatory presentations that we find in these diseases. One of the hallmark symptoms of COVID-19 is ageusia, or loss of taste. Now, does this have anything to do with the microbiome? We're not exactly sure, but we do know that the spike glycoproteins that bind to ACE2 receptors, which are well known for their location in the lungs, are actually very abundant along the gut brush border cells, as well as a certain subpopulation of taste bud cells. So this provides a potential mechanism for local and systemic inflammation and gut barrier dysfunction. Ulcerative colitis and Crohn's disease, also known for dysbiosis, are associated with a decreased taste sensitivity in all tastes except for sour. Up to 84% of patients undergoing chemotherapy experience some type of taste change, and these patients are also known to have a disrupted microbiota. Bariatric surgery is a pretty invasive procedure that changes the anatomy and function of the GI tract, effectively changing the gut microbiota too. It's found that taste sensitivity to sweet and fatty stimuli appear to increase in these patients post-op. For FMT, there wasn't a specific study on taste preference. However, glucose intolerance seems to be fully transferable to germ-free mice through FMT. Diet is well known to be the major modifier of gut microbiota, so it's no surprise that taste preference and nutrient sensing are also shown to change with the habitual consumption of artificial sweeteners and vegetable-rich diets. Anorexia nervosa and obesity patients each exhibit characteristic microbial signatures and are found to have changes in taste perception as well. So with this project, we help paint a picture on how the microbiota might be influencing taste perception. The more we learn about this relationship, the more we can help our patients curb their overeating on the one hand and improve taste qualities in disease conditions on the other. Thank you.